Now that I've given you probably more information than you ever wanted to know about photosensitive cells in the back of your eye, let's turn our attention to the cells that form the final layer and that ultimately have their axons form the optic nerve leaving the eye, the ganglion cells. Now, the ganglion cells come in two information processing flavors. As I've mentioned many times, visual processing begins even before the ganglion cells, and certainly it's occurred by the time the ganglion cells leave the eyes. And in fact, there are already two types of cells that exhibit a specific sort of feature extraction called center-surround antagonism. On-center cells exhibit an excitatory response to light in the center of their receptive fields and an inhibitory response to light in the surrounding area of their receptive field. And so the receptive fields have this structure to them: inhibitory outside, excitatory inside. Off-center cells exhibit an inhibitory response to light in the center and an excitatory response to light in the surrounding areas. And that gives them a receptive field structure like this: excitatory on the outside, inhibitory in the inside. Now, researchers determined that cells that feed in to the receptive field for ganglion cells feed into both on and off-center ganglion cells. And so this allows populations of ganglion cells to detect light intensity contrasts and rapid changes in light intensity across the visual field. You might wonder how researchers ever found out about the light sensitivity of rods and cones or the information extraction properties of ganglion cells as they're leaving the back of the eye. And this diagram and picture depicts this. Right? This diagram shows the typical setup. Here you've got a capillary pipette that's attached to an individual nerve. In this case, it's a ganglion cell. And this is the front of the retina. This is the back of your retina. Right? And they have an animal, sometimes a toad for rods and cones, for example, or a cat. They get access to the retina. They attach pipettes to individual cells. They project something onto the screen, and they observe the cell response once they identify the area in the visual field that the cell is sensitive to. Nowadays, the apparatus looks a lot like this. And now I want to go back and show you that video of the work by Hubble and Wiesel on receptive fields and the properties of individual cells. And we're going to be looking at an on-off cell.